Good morning to one and all present here. My name is Piyush, Assistant Professor, Department of Pharmacology, Faculty of Pharmacy, MS Ramaiah, University of Applied Sciences. I would like to tell all of the participants to please unmute yourself during the webinar. They can just put their mic off. Whatever queries they have, just they can write it down in the chat box. And after the session, definitely each and every question will be entertained by sir. Shall I start, sir? Good morning to one and all present here. On behalf of Department of Pharmacology, Faculty of Pharmacy, Ramaiah University of Applied Sciences, I, Piyush Vats, Assistant Professor, Department of Pharmacology, extends a warm welcome to all of you for the webinar. This webinar will emphasize on various techniques in network pharmacology, an era in drug discovery process. It's my privilege to introduce to you our eminent speaker, Dr. Anoop Kumar. Dr. Anoop Kumar, currently working as an assistant professor in the Department of Pharmacology, Delhi Institute of Pharmaceutical Education and Research, DIPSAR, Delhi Pharmaceutical Sciences and Research University, DIPSARU, New Delhi. Earlier, he worked as an assistant professor in the Department of Pharmacology and Toxicology, National Institute of Pharmaceutical Education and Research, NIPER, Raebareli, as an officiating head and associate professor in the Department of Pharmacology, ISF College of Pharmacy, Moga, Punjab. He has also worked as research scientist in the Medical Affairs and Clinical Research Department of Sun Pharmaceutical Industries Limited, Gurugram. As well as he also worked as Translational Health Science Institute, THSTI, Faridabad. He has completed his PhD in Pharmaceutical Sciences from Birla Institute of Technology, Mesra, Ranchi as a DST Inspire Fellow. He is working as an expert in various bodies like Board of Studies of the Clinical Research Program of IKG-PTU Jalandhar, Punjab and Drug Discovery Hackathon 2020 joint initiative of AICTE, CSIR and Government of India. Recently, he has also been included in top 2% scientist list released by Stanford University. He has authored more than 100 research and review articles and 20 book chapters in the International Journal of Repute. Currently, his current H index is 26 and I-10 index is 63 with citations of 2613. He has also filed 4 Indian patents. He has guided 38 postgraduate research students and 2 PhD students. He has also edited 3 international books. Recently, he has also joined as associate editor in the Health Sciences Review Journal of Elsewhere and Acta Neurologica Scandinavica Journal of Ville. He has currently he is working as member secretary of Human Ethics Committee approved by CDSCO and treasurer of ISPOR India chapter. His lab also gets funding from DST Serve SRG 30 lakhs, ICMR Mission Project 1.3 crores, ICMR Investigator Initiated Project 65 lakhs and 1.2 crores, National Innovation Foundation NIF India 10 lakhs and IIT Delhi 10 lakhs. His research interests are in the following areas like drug repurposing, meta-analysis of clinical trials, network pharmacology, pharmacoeconomic studies, signal analysis and pharmacovigilance, as well as his research area is there. With this brief introduction about our speaker, I would like to welcome you all again to our webinar, Network Pharmacological Techniques, Next Generation Medicine Research Mode. Now, it's my privilege to welcome Dr. Anoop Kumar to take over the session ahead. Thank you so much, sir.
Thank you so much, Piyush, for your kind introductions, and I hope I am audible.
Yes, sir. Yes, sir.
Right, okay. So, first of all, I would like to thank Dean, Faculty of Pharmacy, Dr. S. Bharat, HOD, Department of Pharmacology, Dr. J. Anbu, coordinators, of course, Piyush, for giving me the opportunity to be part of this wonderful event. So, for next 40, 45 or 50 minutes, I am going to talk specifically on what actually the network pharmacology is, and if someone of you would like to use network pharmacology in your field, so how you can use it, what are the different steps, and what work in our lab we have done so far with respect to network pharmacology. So I am going to share my screen. Please confirm when it's visible. Yeah, I hope it's visible to everyone.
Yes, sir. Yes, sir. It's visible. Please
Okay,
proceed, sir.
great. Thank you. Thank you for confirmation. So as I said, network pharmacology, if you see the literature, you will find, if you just do the PubMed and just write the word network pharmacology, you will uh, find lot of research papers which are published from Chinese researcher, and they are using these network pharmacological approaches in their traditional Chinese medicines that they call TCM. With respect to India, now some research papers are coming up from few labs. Network pharmacology is something which will help you to predict the possible mechanism of not only the new chemical entities, but for Ayurvedic formulations, for herbals. You can use these techniques for drug repurposing and so on. So as I said, for next 50 to 55 minutes, I am going to give you overview about what actually it is and what are the steps if any one of you would like to use in your field. So how to do it. So content is, as I already covered, these things I am going to cover in this presentations. But before network pharmacology, you all knows pharmacology is the study of drugs, and there is no, I strongly believe that there is no alternative for the basics. So basics are same everywhere, whether you talk about artificial intelligence, network pharmacology, machine learning. If your basics are not good enough, you can't use these techniques or tool in a better way, right? So just whenever, if you are planning to do a network pharmacological approaches, just in particular area, just read about their signaling, their pathways, then you can better understand whatever the computers will give in the terms of output. If you don't know about signaling in particular disease, and you would like to use that network pharmacology logical approaches, for example for diabetes, and if you don't know how glucose uptake, what are the different signaling going on in the diabetes, how it altered, and then you can't predict the possible where your molecule can hit, and if your molecule is going to hit that particular target, what effect you can absorb. So, this is just a simple slide where you can see there are two things when we talk about pharmacology. There is a drug actions and drug effect. So drugs acts on particular target, and target can be anything. Target can be receptor, target can be enzymes, target can be other things as well. But most of the drugs, about 80% of the drugs shows their actions by acting on the receptors. So when these acting on or whatever the signaling going on, ultimately the signaling will produce some effect, and that effect is visible. But what is the story behind it, that is the invisible, and that we call drug actions or mechanisms by which drugs showing their response. So as I said, most of the drugs, approximately 80% of the molecules are showing their response by acting on the receptors. So just whenever you are planning to do network pharmacology, just revise these receptors and their signaling mechanisms. So I hope everyone who has joined here, they knows there are a category of, these are the category of receptors. There are nuclear receptor, receptor kinases, ligand gated, GPCR, and how this signaling going on. Like in GPCR also we have GS, GQ, GI. If some drugs is attaching to the ligand binding domain, and it could results in the activation of GS, and GS could results in the activation of adenyl cyclases. It will break down the ATP into CMP, and increase CMP will do the phosphorylations of calcium channel if we talk about heart. So what I mean to say, I am not going in detail about the signaling, but what I mean to say is, whenever you are planning to use network pharmacological approaches, try to read the signaling pathway in much more detail. How there, if there is a phosphorylation, so phosphorylation of what thing? If there is a formations of something, like if there is a phospholipase is activated, they do the degradation of PIP2, that is the phosphoinositol diphosphate. I think pharmacy people know about it, and then it result in the formations of two molecule IP3 and DAG. So you have to know where the IP3 will act when it produce inside the cells. It has a receptor, smooth endoplasmic reticulum has a receptor for IP3, and smooth endoplasmic reticulum is a storehouse of calcium, and it do the it interact with actin and myosin filaments, and ultimately results a contraction. There are a means well-known mechanism by which these our ventricles contract, our smooth muscles contracts, like that. So so you read about these pathways before doing the network pharmacologies, because if you are not updating your knowledge with respect to pathways, you can't understand the results, and you will do the mistakes when you are doing the interpretations of the network pharmacological results. So, if you talk about history, there are many molecules which are available in the market, and most of the molecules are on single drug and single target concept. Right now, many researchers are producing more and more selective blockers so that your adverse effect can be minimized. But practically, if suppose you are on beta 1 blockers like drugs, so is that drug is going to block only the beta 1 receptor which are present on the heart? No, similar receptors is also present all over the body. Beta 1 is also in the prostate. So it's practically it's very difficult to produce very much selective compounds, but of course we have produced, but the undesired binding of these molecules could result in beneficial as well as the adverse effects, right? So for several drugs already on the market, as I said, population based study failed to show patient relevant benefit, and our preclinical animal model of disease can often only mimic the symptoms without any evidence that mechanism causing the symptoms in the animal matches the human disease. So that is the problem with our animals model, and therefore we lack the mechanistic understanding of the cause of disease. So every time if some student asks us why we are not able to cure the disease, our science is so much advanced, still we are just controlling most of the disease, we are not able to cure the disease. So why it is so? Because reason is because in the most of the diseases we don't know the exact etiology. Like for example, hypertension, primary hypertension, and 95% cases of hypertension is of primary hypertension. And if you talk about etiology of this primary hypertension, you will find that it's unknown. You are assuming that various factors such as environmental pollutants, this that will play a significant role in the development of hypertension, but you don't have a exact reason. Of course, if a patient is suffering from a secondary hypertension, so of course we know what are the reason, and if you cure that reason, you can cure the disease, right? So problem in most of the diseases is we don't know the exact etiology, and unless and until we don't know the exact etiology, we can't cure the disease. We can manage, we can control. We know if blood pressure is increased from up to this levels, so it could do the damage in the various parts of the body. So we have to take the drug, and we have to take it for a longer time, for the entire life, to control that blood pressure. Because if you are not using the medicines, if you are skipping the medications, your blood pressure will be increased and it do the damage. So the question is obvious, why we are not able to cure the disease? The reason already told, because we don't know the exact etiology. जब तक whenever means जब तक आपको reason पता नहीं होगा, how you will cure the disease, right? So network pharmacology to could help us to understand what is the possible mechanism, or what is the possible etiology behind this disease. That is also the one of the application of network pharmacology. And as I said, many drugs, so this is just I have written, they have various drugs in the market, and these all drugs for the hypertension they are managing. For diabetes, these drugs are doing the management. Obesity, these are doing the management, not the cure of the disease. And reason I already told. So network pharmacology can help to some extent. I will not say that by using the network pharmacology you will get to know about exact etiology and you can cure the disease. No, it will help to a certain level, and then you have to do the animal experimentation. So there is a disease network. When we talk about network pharmacology is devoted to understand drug pharmacological mechanism in the network perspective, and it's a emerging discipline useful in the drug discovery which combine. So to do the network pharmacology, you should have a basics of genomics also. If you don't know the differences, chromosome, genome, genes, so you will get a trouble when you are doing the analysis of the data which you will get from the various databases. System biology, chemistry, you should also know, because we are integrating network pharmacology with molecular docking and dynamics. So to do the molecular docking, you should have a chemistry knowledge as well, which functional group is responsible for the hydrogen bonding interactions, which could contribute for the van der Waals interaction, which could contribute for the electrostatic, and so on. So that knowledge is also required. And of course you should be you should have a skill to use the computational biological tools as well. So it's a new frontier discipline of systematic drug research in the era of AI. And means AI is evolving field, and month by month or even day by day, there are a lot of things are coming up. So it's up to you how you are using these techniques in a better way to provide the solutions for unsolved problems in the field of pharmacy or medicals. So globally, network pharmacology now is also regarded as a next generation's medicines research mode. So popularity of network pharmacology, as I said, if you just type in a PubMed network pharmacology, just click on the search, you will find how many articles year by year are publishing by the researcher across the globe with respect to network pharmacology, right? So just do it. You will find year by year number of studies has been published so much, and when you do little bit more examinations, you will find that most of these network pharmacological approaches researchers are using to predict the possible mechanism of herbals or possible mechanism of these traditional Chinese medicines and so on. So there are a number of, body is a complex machine, is a one of the complex machine. So you knows there are a number of signaling pathways are going on. So you can't understand all the signaling pathway which are going on in our body, but whatever the area you would like to work in, that particular area you should know each and every detail about the signaling pathways. Then only you can means you can do it in a better way, right? If someone is planning to use the network pharmacology for those molecules or for anti-cancer, so and particularly the apoptotic signaling pathway, so first you have to know what are the apoptotic signaling pathways, how the signaling pathway undergone, what are the intrinsic, what are the extrinsic, how the oxidative stress increase oxidative stress, what it going to do, it going to alter the mitochondrial membrane potential, then mitochondrial altered mitochondrial potentials that could result in the release of pro-apoptotic and apoptotic proteins. So you have to know what are the apoptotic protein in the mitochondria, what are the anti-apoptotic. So BCL2 is anti-apoptotic, BAX, these are the apoptotic proteins. There is a balance. So whenever any molecules, if it's going to increase the expression of BAX and decrease the expression of BCL2, so it result in the activations of apoptotic signaling pathways, and pathways can be caspase dependent, can be caspase independent. Caspase independent pathways, there is a apoptotic inducing factors molecule, endonuclease G, cytochrome C, APAF1 and caspase 9 for the caspase independent pathway, and finally result in apoptosis. I am just telling it a little bit faster because it depends upon your area in which you are planning to work, right? So that's why I am just keeping it in a fast mode. So whatever area you have picked to use the network pharmacological approaches, just read research paper not related to network pharmacology, research paper where molecular signaling mechanism has been explored. What signaling, which protein is phosphorylated, then what happens, and expression of that particular kinase is what it's going to do, and ultimately what the effects you are getting, right? So that's important. Like that, I have worked on apoptotic signaling pathways, where we have explored the apoptotic signaling pathway induced by delta methrin. Delta methrin is one of the pyrethroid insecticide which is widely used due to toxicity of organophosphate, right? It is a derivative of pyrethrin. Pyrethrin is a natural insecticide, but it's a photodegradable. So scientists have created a pyrethyroid, and deltamethrin is one of that. And these signaling we have explored computationally as well as experimentally. We have conducted the Western blot, flow cytometries, and RT-PCR to see how this signaling is going on inside the cells when cell is exposed to these particular molecules by doing the primary animal cell culture of thymocytes and splenocytes. So I am not going in detail. Again, another signaling which is important to understand is immune and nervous. These immune system and nervous system are one of the system which are in connected with each and every part of the body. So if you are talk about brain, immune system is also there. If you talk about kidney, immune cells are also there. If you talk about nervous system is also everywhere, right? So you have to understand these both systems in a much more detail about their signaling. Signaling, with respect to signaling, you know, that's the difference. Everyone knows the immune system is two types: innate immunity, adaptive immunity, and this that. But you have to know, you have to understand when your innate immunity, when your phagocytosis process is unable to kill the pathogens, then how this pathogen is hand over to the adaptive immune system. So there is a handover mechanism and there is specialized mechanism, you know, to hand over the pathogen which is not killed by the innate signaling. And in innate signaling also there are a sensor, you know, there is a sensor like toll-like receptors that we call PRR, pattern recognition receptor. They will sense whether it is self or non-self, and then it will result in activation of toll-like receptor dependent signaling pathway or independent. And finally it's going to activate your MAP kinases, NF kappa B, MAP kinase and NF kappa B will act on the nucleus and do the transcription and translation process by which it will release the interleukin. That is the one mechanism by which it will innate immune system kill the pathogens or exogenous substances. But when your innate immune system is not able to handle, so that particular exogenous substances has to hand over to the adaptive immune system, and there is a specialized mechanism. I am not going in detail, but you read about it. There is a MHC molecule, major histocompatibility complex. There is a co-stimulatory molecules, and then only it will be recognized or identified by the adaptive immune system, and then adaptive immune cell like T cells will undergo for the clonal expansions, and number of T cells will produce, and result in release of interleukin, TNF alpha, interferon. If your exogenous substance is virus, so obviously your interferon will be released, and simultaneously these cytokines will activate your B cells, and B cell will produce antibody against it. So I am just giving an overview, but you have to be read it much more detail. How dendritic cells, how MSC molecules are also MSC1, MSC2. Helper T cells are also TH, cytotoxic T cell, T regulatory T cells, and so on, right? So that you have to be understands. And inflammation is another thing. You know, in most of the disease, inflammatory signaling pathways plays a major role. There are two things whenever we ask the students, if not ask, whenever student tell us we forget each and everything. So I always say that write about inflammations and oxidative stress. In most of the disease, inflammation and oxidative stress play a significant role in most of the disease, right? So importance of network pharmacology is you can use these approaches to identify the potential drug candidate. You can use these approaches to understand the drug mechanism. You can use these approaches to understand the drug-drug interactions by studying their signaling, and you can also identify the possible mechanism of adverse drug reactions, which we are doing in our lab to identify the novel signals. And after identification of novel signals, we are using network pharmacological approaches to predict the mechanism, and then integrating with molecular docking dynamics and experimental techniques. So now we talk about simple networks. So in network pharmacology, you might have seen these type of circles and these type of lines. So circle is we called a node, and node can be gene, node can be protein, node can be metabolite, right? So it depends upon if you are doing gene network, protein network, or metabolite networks. So your circle will represent that part, and that we call node. And line connecting these nodes, these we call edges, and these lines tell you about the relationship between the nodes, between the two genes, between the two proteins, between the two metabolites. So it's a very simple network to make you understand what the circle represents. Circle is a node, and these are the edges. If they are close to each other, then we will say that the relationship between A and B is much more as compared to B and C. So this is a simple network. So important terms related to network you have to read before doing the network pharmacology. As I said, network is a well computed mathematical representation of various connected node and edges. So what the network is? Network is a computed mathematical representation of various connected node and edges. And how is a node with a high degree? And what is degree? Degree is the number of edges connected to the node. It's very simple, right? Betweenness, the number of shortest path that go through a given specific node, that is we call betweenness. And these all parameters we are calculating when we are doing the analysis of the network. And what do you mean by shortest path? It's a minimum path between the two nodes in a network. So you can see from these pictures, for better understanding, you can understand what is the module, what is the hub, what is degree, what is betweenness, and what is the shortest path. So these terms you should aware about it before doing the network pharmacology. Otherwise, software will give the result, but you are not able to analyze it in a proper way. So biological network, human body, we are assuming at a steady state and natural equilibrium state is at healthy state, and a stable network with specific topological structure and certain network properties, and unstable network with different, if we call it unstable network when there is a different topological structure and network properties. So network pharmacology is proposed based on the theory of network biology, biological balance, and it provide a new ideas for the drug discovery as well as understanding the mechanisms. So types of network, as I already told, depending upon the node. If your nodes are gene, then you will get it called a gene regulation network. If it's a protein, protein network. If it's a signals transduction network is also there, metabolic network. So depending upon the nodes. So common databases and tool which we are using in the network pharmacology are, there are so many tools which are freely available as well. There is a Swiss target predictor, and there are tools which are freely as well as commercially available. Same tool is available in a two different form. These are Swiss target predictors, C database, Stitch database, and drug binding database. These are the databases we are using for the identification of the target of the compound. So this is the first step in the network pharmacology. You have to identify the targets of the compounds for which you would like to explore. So for that one, maybe the question arise in your mind. I have shown you these are the databases which we are using for the identification of target of compound. So question might be arise in your mind, we have to use all these databases, or we have to, or one database is enough to identify the target of compound? My answer would be, you have to use all. You can't say that you have used only the Swiss target predictor for identifying the target of compound. You can see some research paper where they have used on a Swiss target, but that is not the comprehensive approach. To do the in a better way, you have to use all the databases. Apart from these databases, if you are getting some additional databases, go for it. Our purpose is to make a comprehensive file first, where you will identify the possible targets of the compound. Apart from these databases, you have to do the data mining of the literature as well. Then that excels will become a comprehensive. Otherwise it will not a comprehensive file, and any experts can raise a questions on your studies as well. Right. So now next thing, maybe your question would be, if we are doing same thing in a different databases, so there is a chances of duplicate, right? If I am putting same compounds, for example routine, I am extracting the targets from the Swiss target predictor, then C databases, Stitch databases, and drug binding databases. So there is a possibility of duplicate, right? Yes, there is, of course there is a possibility of duplicate. You have to put in Excels and just identify the duplicate and remove it, and note these number. When you are writing a manuscript, you have to mention that I got these many targets from this database, these many targets from this, and from this, and then finally I have detected these many are the duplicates, and this is my comprehensive file for the identification of targets of the compound. Second is, targets involved in the disease. So it depends upon in which area you are working. If you are planning to work in a Alzheimer disease, so of course you have to be you have to extract the targets which are involved in Alzheimer disease. If your research interest are diabetes, so you have to be identify the targets which are involved in diabetes. So from where you will get these targets? So there are, again we have a databases like DGNet. DGNet is most widely used databases for the extractions of the target which are involved in the disease. But gene card is there, OMIM is there, HPO is there, Malacard is there, and again you have to use all these databases. Don't use single one. Of course maybe you will get a publication in journals, but later on if you are doing PhDs or something, so experts can raise the questions why you have used a single database. The reason is, particularly if you are using a freely available databases, the disadvantages with freely available databases is there is no fixed timeline to update these databases. So before using that databases, you have to read about these databases as well, right? So you have to use these. Then next step is you have to identify the common targets, and these are the many tools. There is a then interaction among targets. There is a string, biogrid, DIP, stage, inect, These are the different tools by which we can get the interactions among the targets. And for visualizations, we are using the cytoscape or creations of the network or analysis of the network. Cytoscape is a well-known softwares in the field of network pharmacology, and beauty is it's freely available. And then we have to do the SR plot. By doing the SR plot, you will get to know what are the pathway which are involved in that particular compounds in that particular disease, right? So as I said before, using these databases, you have to read these databases carefully. You know, you have to see what is the basis of this database, means how this database has been created, and what is the how these targets has been identified, and when this databases has been updated last. So you have to be see these all informations, you have to read about it. If you talk about Swiss target predictor, so you know these targets are ranked according to 2D and 3D similarities, and 2D similarity is quantified by Tanimoto coefficient between the fingerprint vector, that will be called FP2, of both the query and screen molecules. And 3D similarity is, you know, month and distance between the electrospace vector, that will be called ES5D, of 20 conformers of both query and screen molecules. So threshold we are setting as 0.85 for ES5D similarity and 0.65 for FP2 similarity. And below such threshold, the known actives are not similar enough to be displayed. And if you are doing manually some changes in these threshold, so that's not the good predictions. You know, that's not a good network. You will identify it, you will create the network, that's fine, but when you are planning to verify it experimentally, you will not get anything. There is a maximum chances you will not get anything, that target will not express. You know, so whenever you are using any databases, use the cutoff. What is the purpose to cut off, on what basis they are giving the values, you have to read it. And probability, if you are getting the p values of 1, that means most probably the query molecule is a part of known active screened molecules. So you have to be see if there is a probability of 1 you are getting. So that's a best prediction. You know, that what does it mean? It means whatever the molecule you are putting in the database, that molecule in the database is already experimentally tested on this particular target. So you are getting that values, it's a query molecules. So this is the web page of Swiss target predictors. It's a freely available. And suppose you are planning to work on zebrafish, you know, and you are after doing the network pharmacology approaches, now you plan to test efficacy or safety of that XYZ molecule in zebrafish, and you have used the Swiss target predictor for the identifications of the targets of the compound. So experts will definitely raise the questions on your protocol. The expert will say that Swiss target predictors providing the data of only the Homo sapiens, mus musculus, and Rattus norvegicus, not the zebrafish. So what is the purpose? How you will extrapolate these results for the zebrafish studies? I hope you all are getting. So you can see selection of species. Why I am emphasizing on this part, it's very important. I have seen many students are doing this network and they are not reading anything, and it will not work. You will stuck somewhere, right? So first, whatever the database you are using, read about it. Every information is there in the database, how it's created, and of course every web that database have a research paper as well, in which if you go through these research article, you will find that what is the basis of creation of this database, how it created, what is the vector, what values they have taken, why they have taken, why this which column values you have to be take further, and what that particular value represent. Many time when we ask the student, okay, you got this Excel file from the database, tell me about what this particular column represent, and they are unable to answer it because they have not read about it, and which column values is more important as compared to this. So that represents you are not reading about that particular database carefully. So you can see in the Swiss target predictor, we can get the this database contain the targets of Homo sapiens, mus musculus, and Rattus norvegicus only. Suppose after this you are planning to do the work on rabbits, so the rabbit data is not there in the Swiss target predictor. And these databases are very easy to use. You know, you have to be add smiley notation of the molecules, or you can draw the structure of the molecule. Just click on the predict targets and you can get it. Then there is a, as I told, there is a other database that is we call C database, similarity assemble approach, and this is database is of free of charge, and this is provided by the certain laboratory in the Department of Pharmaceutical Chemistry in the University of California. And what they have mentioned disclaimer in this database, if you go through it, they have written that you can use this database at your own risk. So what does it mean? It means if suppose your molecule is very much confidential, you are working on a new chemical entities, and you are putting your molecule in these such kind of freely available databases, they will not take the responsibility, because your molecule can be means uh, anyone can be tracked that molecules, right? So they are not taking that responsibility. They are saying it's a free of charge, but it's your own risk. If you plan to use for your molecule, you can use it, but we are not taking the guarantee about the data confidentiality on this public service website. I hope it's here. That's why many researcher in network pharmacology, they nowadays, particularly pharmaceutical companies, they are using commercial databases instead of freely available. But of course, most of you are in academia, so in academia we can do it. But if you have a very much confidential molecule and you have not filed the patent yet, so my suggestion will be first file the patent and then apply these network pharmacological approaches, right? So this is the C web page, and read about it in much more detail. It's not possible to tell you each and everything about these data sets in a 1 hours. So this is another database stitch, and this is a website for the stitch and database of known and predicted interaction between the chemical and proteins, and it contains data from the genomic context prediction, high throughput lab experiments, and automated text mining approaches. And you can see how many proteins data and organism data is available, and this stitch is available freely as well as commercially. And combined score, it give you the combined score, and it's computed by integrating the probability from the various different types of evidence. So this is a web page of the stitch. Then there is a binding database, it's a based at the School of Pharmacy and Pharmaceutical Sciences at the University of California, and it's a public web accessible database. And this is the web page of the binding DB. You can read it in much more detail about drug binding DB databases used for the identifications of the possible target of the compounds. Then there is a DGNet. DGNet is particularly for the target which are involved in that particular disease. So it integrates disease and gene related information. So you can see how many numbers of genes, disorder, trait, clinical and abnormal human phenotypes, variant disease association are available in the DGNet. DGNet is freely available, but there is a DGNet Plus. DGNet is commercially available, and this DGNet Plus have obviously it's a update. DGNet Plus have a updated data, whereas DGNet is not updated frequently. So this is a web page of DGNet where you can get much more information of the DGNet. And then there is a gene card. So it's established in 1997 by Crohn Human Genome Center of the Institute of Science in Israel, and comprehensive and easy to use human genome annotation database. And it provides the relevance score. So you have to see what cutoff values you have to report and how this relevance score has been created in the gene card. So this is the gene card web page where you can use it. But before using it, read about it. Read the research papers which has been published on gene card. You will get much more interest about these techniques. Then there is a OMIM database. OMIM database is a comprehensive database on human gene and hereditary trait and relationship between disease phenotype and genotype. And best base, it's updated daily. And this database was initiated in 1960 by Dr. Victors as a catalog of Mendelian trait and disorder entitled Mendelian Inheritance in Man. That's why its name is OMIM. An online version of OMIM was created in 1985, and you can see it's created by the prestigious institute like John Hopkins. This is the OMIM, and 50 years of OMIM has been completed, so it's a updated database, and they will provide you date as well when it's last updated. So you will get to know that when it's last updated. And then there is a HPO. HPO is a human phenotypic ontology database, and it's 2007. It's created in Carrier University Hospital in Berlin. Provide information on medical related phenotype. And NIH supported project that integrate, align, and redistribute cross-species gene, variant, and phenotyping data, and available on this website. This is the web page of your human phenotypic ontology, where you can get more information about it. Then there is a Malacard. Malacard is a human disease database, integrate both specialized and general disease list, including rare disease. Because if you see earlier databases don't have a rare disease data. So if you are planning to work on rare disease, whatever area you have, and you are planning on the rare disease, so if you are using the data of gene card or DGNet only, so obviously it will means question will be raised on your protocol, on your studies. You have used these databases, but these databases does not contains the data of the rare diseases. So how you will defend it? So that's why again I am repeating, whatever the databases are available, read about it, which databases, and then think about it, in which area I am working and which databases I have to be extract the informations. But thumb rule is you have to use all the available databases, because if you are using the freely available databases, right? So this is the web page of Malacard. Malacard is a human disease database, as I said. So you can explore it in a much more detail about Malacard. Then there is a data mining approaches. These are the databases, but you have to do the data mining as well, data mining of literature as well. The reason is, suppose whatever the database you have used, it's updated in 2020. So now we are in 2024. So there are a 4 years in which maybe new target has been identified. So how you will get that information? You have to do the data mining of the literature. Then you have to add these targets in the Excels. Then your Excels will become a comprehensive file of that. So you have to apply data mining, where you have to be apply specific systematic approach to identify the literatures. I am not going in detail, but maybe many of you knows how to do these. By using the PubMed, you have to identify your mesh terms, then use the proper Boolean operators, and then make the search strategies. And then you can, in the advanced versions, you can do the filtering, and then you can do the screening. And nowadays we have a AI based tool as well. Rian is a will help you to speed up the process, where you can do the blinding, you can do the inclusion, exclusions, everything you can do it by using the Rian. Then there is a next step in the network pharmacology. After preparing the comprehensive file, you have to do the string, and before string you have to identify the common target, and then you have to see the string where you will get the interactions among the common target. So there is a cutoff values is 0.7, and you have to know it to give the combined score, and on the basis of probability from the different evidence, you will get these cutoff values, and this is a string web page. Then there is a biogrid is another one, and all data are freely provided. This is the biogrid web page. And there is a DIP is also important, database of interacting protein, where you can check the interactions. This is the database of interacting proteins. Then there is a intact is another one, intact molecular interactions database, which you have to use. And of course there is a VANI, where you can identify the common target, and after that you can create the network by using the cytoscape. I think many of you aware about cytoscape. And then you have to do the CAG, how to fully display the biological information contained in the cell with the help of computer. In simpler way, we called it a CAG, how to fully display the biological information contained in the cell with the help of computer. So CAG is Kyoto Encyclopedia of Gene and Genome. If someone asks you expand the CAG, so you have to say Kyoto Encyclopedia of Gene and Genome, and established by University of Bioinformatics Center of Kyoto University in 1995, and need to predict complex cellular pathway based on the informations in the genome. Because earlier whatever I have shown you, you can identify the targets, but you can't identify the pathway. In one pathway there can be involvement of multiple targets, right? So steps I already told experimentally how we can do it. So step one, I already told you how to identify the targets of the compounds. So to identify the target of compounds, many web server will ask you to put the smiley notation. So from where you will get the smiley notations of the molecule, you can get it from the Campbell, you can get it from the PubChem and other tools, and then you can paste in the queries, and then click on the predict targets. You will get such kind of results. Here you can see the export results. In export result, you can click on the CSV. CSV is the Excel format, and you will get the targets in the Excels like this, and where you will get the uniprot IDs, Campbell, target, probabilities, and known activities 3D by 2D. So you have to understand each and everything. If there is a known activity 3D by 2D, so what it represent 2 by 2, AAA, what it represent AAA, what 0.43, what is this? So everything you have to know, then only you can do a network pharmacology in a better way, right? It doesn't mean someone have told you, okay, these are the target and these are the common name. forget about this. No, that's not the way. You have to know each and everything, right? So next step is identification of the disease targets. You can use the DigiNet where you can create your login ID and passwords. So where you can add the name of the disease and then you can extract the information in the Excel by summary gene disease associations like this, and you can get gene IDs, the disease ID as well. Then the next step is, but again I am just showing you from the one database, but you have to do it from the all the database. Then interaction among target string, just type in a Google string where you can add the common target and you can get the interactions among the targets like this. And from string you have to download the file of CAG as well, and that CAG, if you see in the note sheet, it looks like this, but you put in the Excels, it will be arranged automatically. All the column will arranged automatically. Next step is, it's very important to do some manual work as well to do the network pharmacology in a better way. So what you have to do is, you will get these signalings, term descriptions in signaling pathways, but you have to identify in that particular disease what are the most important signaling pathways. So that you have to be highlight, okay, this is a well known pathways, and on this particular pathways there are already drugs in the market for the treatment of this XYZ disease. So you have to be identify all the possible target which have a role in that particular disease, and then you can do the little bit sorting. You can do the text to column. These are the matching protein in your network level, so you can do the text to column, and then it can be arranged automatically. Not automatically, you can put in a such a way that, like you can see map kinase signaling pathways. These are the pathway and these are the targets which are involved. So in one pathways there can be multiple target, right? In map, if you see the map kinase signaling pathway, you will find there are number of targets, there are number of proteins which are involved in that particular pathway, right? So that you can do it, and then you can put the targets which you have identified a common target, and you can apply the VLOOK formulas. I hope many of you are aware about VLOOK formula in the Excels. We are using these VLOOK formula for multiple purposes to identify the common targets, and if you are not means familiar with that, so you can use the Benny tool as well. So where you can identify the drug target, put the drug target, and here disease target, and these common target you will get from the Benny tool. And then if you planning to do by the VLOOK, so this is the formula for the VLOOK, where you can one side you can get the targets, node 2 targets, and these are the particular target which your compound can be hit. So you will by putting the VLOOK you will get to know which are the target which my compound can hit in that particular disease, and that is your objective. You want to see which target my molecule can hit in this particular disease, right? So next you have to create the network. For creation of network you have a cytoscape. So cytoscape is easy to use. You have to just upload the files from the compound files and then network these pp files, and then merge these networks, and there is a merge options, and then you will get these nodes and you will get the networks. And then there is a cytohuba is a app inside the cytoscape where you can calculate the various parameters. It will give you the values of what parameter, betweenness, these type of things you will get it. Then you can identify the most top target as well, because by doing the network you will get there are a number of targets. So practically it's not possible to do the Western blot or other techniques to measure that targets. So you have to identify which are the most important targets. So there, by using the cytohuba you can put the filters. You can keep here top 10, top 5, and that results the software is providing depending upon your betweenness, depending upon your shortest path. So you will get these. So, and there in the layout you can if you go through a network pharmacology research paper, you will find some researcher are presenting it into a circle, some are representing in a some other way. So there are different ways to present your results. So it's up to you what kind of layout you would like to give. Then after that, as I said, CAG you have to do, that is the enrichment analysis. For to do the CAG there is a gene enrichment pathway enrichment bubble is there. So by these SR plot you can use the enrichment bubble, and also you can use the these particularly SR plot. But apart from SR plot you can also use the Shiny Geo for the KEG enrichment pathways. So these are the these are the Sinogeo. Sinogeo is also freely available, which you can use to identify the pathways. Then next is, as I said, by network pharmacology, you get to know about which target can be involved in that particular disease, not particular disease, which target your compound can hit in that particular disease. So after that, you have to check the interactions, because network pharmacology will not tell you about how your drug molecule is interacting with these identified targets. To do that, you have to do the docking studies. So I think everyone nowadays is aware about what actually the docking is, what are the different steps. You have to prepare the proteins, prepare the molecules, then you have to do the you can do HTS, SP, XP depending upon the number of molecules, and then you will get the scoring functions, target selections. So I hope you know how to select the targets, what are the different parameters to select the targets, depending upon resolution value, depending upon mutations, depending upon method of crystallizations which was used for the crystallizations of that proteins. So you know, when we are putting sometimes in the search item name of the target, you got for the one target you will get multiple PDBs. So now there is a confusion which one I have to take. So these are the parameters on the basis of that you have to take. In our lab we are using the Schrodinger. So I have just added a few screenshot of the Schrodingers which we are using, Mastro, and for the target preparations, grid generations, and then we are doing these docking studies, and then we are doing the calculations of MMGPSA, that will for a binding energy of protein ligand complex estimated by using molecular mechanics generalized bond surface area, and this is the equations for it. Maybe many of you knows about it. Then next step is after docking, you know, docking will not tell you about whether your molecule is stable inside the active site or not. To do that, we are doing the molecular dynamic studies. And for molecular dynamics, GROMAC is freely available, but of course, if you have a Schrodinger license versions, you can use the Desmond, and you can do it in a more easier way. But of course, many researchers are also using the GROMAC as well. GROMAC is freely available. Just go through the GROMAC tutorial, and these are the steps which we are using in the which we have done these molecular dynamic studies by using the GROMAC tool. There is a other software as well. And you know, this molecular dynamics is depend upon Newton's second law, whereas docking is depend upon your law of thermodynamics, delta G is equal to delta H minus T delta S. I hope you all knows what are the principles. And acceleration is equal to change of the velocity with respect to time, and according to that we are checking the movement. We are giving the simulations for 100 nanosecond, 200. Nowadays journal are asking to simulate for 200 nanoseconds, some are asking for 300 to see the exact picture of your ligand in the active site of the target. So Desmond is easy to use to do the MD, and this is a system build up step by using the Desmond, and then you can upload and you can calculate the parameters. And in MD we are calculating the RMSD, that is root mean square deviation. So you can see that these molecules we have repurposed, identified and repurposed for the bacterial infections. Currently experimental study is going on, and we are getting a good results for some molecules. That is RMSD, we are calculating RMSF, then SASA, then number of hydrogen bond, radius of gyrations, and post MD binding free energies. These are the few well-known parameters when we talked about the molecular dynamics. Apart from that, many journal will ask you to provide the PCA, that is the principal component analysis. So we have done the principal component analysis as well. Then there is a free energy landscape as well to get the real pictures about the ligand in the active sites. Then challenges, challenges with respect to network pharmacology is insufficient data, particularly when we are using these freely available tool. As I said, many tools are not updated time to time. And what is how to handle that challenge? As I said, you have to do the data mining of the literature. Data mining of the literature in a better way. You will get a new target as well, and put these targets in the Excel file and make it comprehensive. The two steps which I have shown you at the first, identifications of the target of the compound and targets which are involved in that particular disease. These are the foundations for the network pharmacology. Next step, whether it is a string, whether it is cytoscape, whether it is a CAG, they depends upon these two steps. If you are doing the mistake on these two steps, your whole network will go in the vain. So you have to be handle this insufficient data. Of course, it's a little bit time consuming, but you have to do it. And then quality of data is because there are many databases, their quality is not assured. So you have to be, that's why I am saying whenever you are using the databases, read about each and everything, how this databases has been created, what is the basis of this data in this particular database, what is its limitations. So all things you have to read it. And then multiple data sources, of course, as I said, multiple data sources are there. We don't have a single data source where we can extract the all the possible targets and then we can do the network. So international standard for network pharmacology evaluation method guidance has been developed, and they have mentioned how to do the network pharmacology. Because problem with network pharmacology is if you go through a research paper, every researcher is using a different methods. Every researcher, some researcher are doing first Vanny tool, then they are doing the CAG, some are doing some steps first and some steps second. So there is not a standard guidelines, how to standard guidelines or protocol to do the network pharmacology. So now it's going to develop and it's going to come soon. That is a network pharmacology evaluation method guidance. Particularly Chinese researchers are working on it. But being Indians, I always feel that we should also create a network pharmacological evaluation method guidance for herbal or Ayurvedic drugs as well. So it's opportunity for us to do that. We are planning to do that. We have make some team as well. If someone of you are interested for that, so you can contribute that method guidance document so that we have a standard document by which researcher will get to know this is the way how to do the network pharmacology. And this is a work which my students have done with respect to network pharmacology recently, repurposing of drugs against hepatic encephalopathy using pharmacovigilance based approaches integrated with experimental verifications. So what we have done, we have we have worked on the hepatic encephalopathy is a one of the major issues, and for that one we have identified a repurposed candidate, new candidate which is not earlier known. Repurposing of the candidate means you have identified such a candidate which is there is no report is available in the literature which show that this molecule can be used in that particular disease. That is we called a novel drug repurposing, right? So how we have done it, we use the post marketing data of the drugs, and post marketing of the data we have extracted from the FAERS, that is the FDA adverse event reporting systems. And then we have do the inverse signals, and by inverse signal criteria we have applied some statistics, like we have calculated the PRR, proportional reporting ratio, reporting order ratios, and then informations components and so on. And then we have identified some molecules, and then we have makes the charts. And then after that, because you have identified a new molecules in that particular XYZ condition, particularly in this case hepatic encephalopathy, so we don't know the possible mechanism to identify the possible mechanism, because it's a new thing which you have identified. So we have used the network pharmacology, how these molecules, identified molecules, could act, could be a promising candidate for the hepatic encephalopathy. So as I said, the different steps. So we have done the Vanny, then string, then we have cytoscapes, then we have molecular docking and dynamics, then we have done the experimental animal studies as well to verify these results. This is the experimental studies protocol. I have not included all the results. Then histopathologies and various parameters we have measured, and we get to know that yes, this drug could act as could act in the hepatic encephalopathy. And this is the signaling which we have identified, and these are the drug amlodipine and carvedilol. So I hope everyone know amlodipine is a well-known calcium channel blocker, carvedilol is a beta blocker used in the cardiovascular disorders, but we have identified their alternate use for the hepatic encephalopathy, and that is the drug repurposing, right? So this is the particular pathways we have explored. It's act on the receptor tyrosine kinase in hepatic encephalopathy. Your endogenous molecule act on this target and going to activate various effector molecules and going to activate PI3 AKT signaling. So I am not going in detail, but you can see these signalings we have identified, and we have identified this carvedilol can inhibit the NF kappa B, it can inhibit the MMP9, amlodipine can inhibit the PARP1. And due to inhibition of these, the cytokines which level TNF alpha, IL-1 beta, IL-6, IL-10 IL-10 is anti-inflammatory, so their level will increase, IL-10 level will increase, and this level is going to decrease. So these we have measured by using the ELISA, and we found that there is a decrease in the inflammatory markers and increase in the level of anti-inflammatories. Then another studies which we have done on the recent studies by using the network pharmacology is a drug resistance epilepsy, and in that one we have identified some natural compounds against drug resistance epilepsy. But before doing these studies, we have performed the SLR and meta-analysis, where we have identified the natural molecules which are showing their promising effect in the epilepsy, not in drug resistance epilepsy, in epilepsy. So based on that one, we have selected these molecules according to their pharmacokinetic parameters, according to their bioavailabilities. We have screened these molecules and then apply the network pharmacology, and finally we have identified the luteolin and silymarin, which could act as a potential candidate for the drug resistance epilepsy. These are the our findings. We have done again whole circles, network pharmacologies, then we have identified the pathways, then docking, then dynamics. But again, first we have understands what is the epilepsy signaling. So means in normally how epilepsy is developed, but is the brain signaling, because brain signaling is something which is very complex. So you have to read much and much more about it, then you can do the interpretations in a better way. So I think time is short. So these are the experimental. Then after these, we have done the experimental studies. We have done the HPLC, where we have measured the level of these neurotransmitters, and then histopathology as well. This is another work which we have done in collaborations with Naiper Raebareli's, where we have where we have the Stenospora cordifolia. We have found that it enhances the immune response in the mice immunized with the Japanese encephalitis vaccines, and we have used network pharmacological approaches integrated with the experimental approaches. So we have done this part means network, and there are some other articles which we have published with respect to identification of novel signals. We have identified a novel signal of Raynaud phenomena with calcitonin gene-related peptide antagonist using data mining algorithms and network pharmacological approaches. In this paper, we have not used we have not done the experimental techniques. We have done up to network pharmacology. So these are some of the studies which I have done recently with respect to network pharmacology, and I would invite all of you, if anyone of you is working on network pharmacology, particularly in the traditional medicines. Yeah, we have a special issues for that, and you can submit your paper to this journal, Health Sciences Review Journal. Although it's there is no impact of this journal, it's a new journal, but we are hoping in upcoming years it will impact factors will come for this journal, and you can submit for the special call. And for special call, there is no APC, there is no article processing charges. It's a free of cost, but your work should be related to network pharmacology in traditional medicines, right? Then another call for paper which me and Dr. Divesh is editing is current research in biotechnology. Its impact factor is 5.6, and we are inviting the studies who have used network pharmacology, but we need network pharmacology with experimental approaches and with respect to natural product drug discovery. If you have such kind of work or such kind of manuscript, you are welcome to submit your paper in this journal under these special issues, right? And next is conclusion is a next generation medicine research more, but most important thing is you have to do lot of depth analysis. Signaling, it's easy to say that it's easy to understand the signaling, but it's very tough. It's very various signalings are overlapping to each other. Sometime kinases phosphorylation do the contraction, sometime it do the relaxation as well. Sometime increase CMP will do the dilations of that particular tissue, sometime it do the contractions, right? So there is means you have to read and rapid evolving field. So I would like to acknowledge my team members. There are PhD students, JRF, DST Serve, and this is our team. We are also working on nutraceuticals, and we have put some molecules in the markets with respect to nutraceuticals, where and we are using this network pharmacology to explore the possible mechanism of nutraceutical, because nutraceutical is something which lacking the scientific evidence. So we are generating that evidence. This is the websites of our web page, and I would like to acknowledge various funding bodies, DST Serve, who from there I got two projects, DST Serve SRG and CRG, ICMR, Sun Pharma, ICMR, 2-3 projects are undergoing. Sun Pharma, where I have learned novel signals or other stuff. IIT Delhi, where I have used the supercomputer facilities of IIT Delhi and completed one project during the COVID time. By using that facilities to we have done some homology modeling for the proteins. And then Naiparai, of course, where I have learned a lot of things. VIT Misras, where I did my PhD thesis. This is my first job after PhD. ISF Moga, and of course my current organizations Dipsaru, Dipsar. So thank you so much for Ramaiah Universities and all the listeners who have listened patiently. Thank you so much for your joining and your time even on Friday. So I am happy to answer if you have any queries. Thank you so much.
Thank you very much, Dr. Anoop sir. Really, it was a wonderful discussion. I have so many doubts. Sir, can you hear me?
Yes, yes, sir. Go ahead.
Yeah, actually, I'm very much thankful to you for sharing your research experience and in a detailed way. You explained about network pharmacology. As you mentioned in your discussion, so in the initially in the network pharmacology, you explained about that various steps involved, right? So
Yes,
in
yes.
that, in identification of targets, using various databases, about around 17 databases that you have explained for
Right,
both, all the steps. So in that,
right,
you very particularly mentioned about that edges and nodes, right?
right.
So in that, my question is actually by using the databases and further into data mining process. So
Mm-hmm.
you have explained about the tools to be used for identification of common targets and also targets involved in the various diseases and interaction amongst the various targets.
Right,
Finally,
right.
you end up with that visualization. So now
Yes,
for just for example, so Swiss target predictor and you have mentioned for Homo sapiens and rodents generally.
yes, yes, yes.
So can you suggest some databases for other veterinary other species of animals for as a predictor? First question.
Yes, yes, sir.
Can you
Apart
please?
from Sys Target OMIM database, where you will get all the species databases, means data. For with respect to diseases, DigiNet and these gene cards are comprehensive. But again, means you are working in any species, but you have to use
Yes,
extract the
the
data
reason is
from the
actually
all
my
data.
peoples are now initiated some work with zebrafish. Is it
Okay.
possible or will you suggest some specific databases
No,
which is
no,
suitable
sir.
for identifying the
No,
predictor
there
in
is no
zebrafish
specific
related?
databases with respect to zebrafish, sir. But
Okay,
you have to use all the databases, and along with databases, you have to extract the data from the literature with respect to zebrafishes from the research
okay,
articles.
okay,
That will help. Of course,
right,
sometime practically when we are doing these exercises, you will get to know that same target is in the zebrafish, same target in rat, same target in the mice, right?
okay, okay,
So that's
right,
overlap. So, but
right, right,
thumb rule is we have to use all the available databases. And for
okay.
zebrafish, there is no specific database is available, but you can get it from the research studies.
Hmm.
And whenever any
Fine,
expert will
fine.
ask you about it, then you have to say, I have done data mining of the literature as well. And from literature, these many these datas I have extracted, and these are the target. These are specifically related to zebrafish.
Okay, and
Because
okay,
sir,
sir.
ultimate sorry to interrupt,
No, no,
ultimate our
please.
objective is to put that molecules in the clinical trial, right, in human,
Yes, yes.
right? So there is a target which will overlap among the species. So we can't say that if I am working on zebrafish, so I have to use only the zebrafish database. No, you have to use all.
Okay.
Right.
Right, and that's in the same way. It's my next question actually. By using this network pharmacology, we can establish the mechanism, possible etiological aspects as well as adverse effects. Also, you mentioned.
Yes, yes, yes.
So, can we use this technique as an alternate method? See, in general, in the clinical side, we are using various other softwares like Pies Index or any other such kind of softwares for identifying the adverse events. So can
Yes,
we
yes.
use this network pharmacological softwares as a alternate tool for identifying the adverse effects in the case of a repurposed drug as well as that existing molecule in the therapeutic use?
Yes, sir. These particularly, this particularly in with respect to pharmacovigilance. particularly the signal that we call signal team. In pharmacovigilance also means there are different teams, and there is a signal team. The signal team work is to identify the novel signal of the drugs.
Yeah.
Means the ADR which is not already known, means that is not they have got in the clinical trial, but when drug reach in the market, they got that ADR, and that is not mentioned in the SMPC of that particular molecule. So that we called it's a new ADR. So when you identify a new ADR which is not earlier get in the clinical trial, so that means it's a new ADR. So of course you don't know what is the possible mechanism behind it. That is the one thing. Another thing is, even though we have a many known ADRs, and but we don't know the mechanism of these. There is a ADR. If patient is getting diarrhea, patient is getting means any ADR, tachycardia, but we don't know the possible mechanism behind it. So we can use network pharmacology to identify the mechanism. But to identify the ADR, network pharmacology will not help. Because to identify the ADR, you need the post-marketing reports. You need the ADR reporting. You need these FAERS database. You need UDRA vigilance.
You mean
is another
we
one.
can identify a specific pathway which leads to a specific adverse effect. So that is the main usefulness of network pharmacology, right?
And by that information, you can manage that ADR. That will be called signal management. Okay, you got to know that this signaling, by activating this signaling, we are getting this ADR. So you can think of how to handle it, because it's not easy to withdraw the drug from the market. So you have to see, okay, this drug is very good, but it's doing this, and by this signaling, so we have to add another molecule or other things to prevent this activation of this signaling. Right, sir.
Fine. And my another last question, sir.
Yes, sure.
Actually, at the end of your discussion, you mentioned for the traditional medicines also, including that other Chinese medicines also, this technology can be used, method can be used for establishing this mechanism and other thing, right? So in that case, so if suppose in our Indian system of medicines, we have a different variety of traditional systems, like Ayurveda, Siddha, Unani, multiple type of systems, right?
Right, sir.
So if suppose if a drug is intended for a multi-pharmacological action, that same drug in some of the Indian system of medicine like Siddha, by simply changing the adjuvants, it is applicable for treatment of various disease condition.
Right.
Is it possible to establish simultaneously the mechanism as well as possible etiological details and also adverse effects for a particular drug which has a multi-pharmacological action by using this network pharmacology?
Yes, definitely, sir. That's why I said a lot of network pharmacology is used in the field of herbals only. The reason is we know we know this herbal is effective, and there is a patient data as well. But when we go at the international platform, they are asking us to provide the scientific evidence, and we don't have it. We are saying, okay, haldi is okay, and it's doing this, that, and we have a patient report as well, but they are not believing on it. They are asking us to provide the scientific evidence.
Scientific proof.
Yeah, so network pharmacology is the one of the technique by which we can generate the scientific evidence, and we can show them this herbal is working, and by through these multiple targets. Right. And unfortunately, means, if you see these Chinese are doing, why they are using these and publishing these papers in a good journals, because their first line treatment is traditional Chinese medicines, and they are generating the evidence by using these techniques. So in India, I think we also have to use these techniques and strengthen our Ayurvedic or herbal systems. Means definitely it will help much more.
Fine, sir. Thank you very much for your patient answering, sir. Thank you.
Thank you so much, sir, for your questions. It's my pleasure.
Thank you. I think other people are waiting with some other question.
Okay, definitely.
Dear participants, your mic is enabled already. Whatever queries you have, just you can directly connect to the speaker, sir. Sir, there is one question also from one of the participant, Dr. K. Sundara. He is asking actually, is okay to perform experimental validation in animal models alone after performing network pharmacology? Means he's asking,
Yes.
is it okay to perform experimental validation in animal models alone after performing network pharmacology, sir?
Yeah, alone means they don't want to do docking or dynamic studies.
No,
Yes,
sir,
sir,
sir, I'm
exactly.
online. I'm online. Is it
Yes, sir.
audible?
Go ahead, sir.
Yeah,
Go
my
ahead, sir.
concern is like since in network pharmacology we are constructing network and we are finding the pathway which is involved, all those things. So ultimately we are working at the gene level. So
Mhm.
doing after this, so should we? Is it animal model alone is sufficient, or should we go for even cell lines also?
Cell line also, sir, means first you can do the in vitro, but again, depending upon if you don't have a facility for in vitro, so you can directly go for the animal studies. Yeah,
Okay.
and particularly when you are going for animal studies, you have to be very carefully analyze your result. You know, sometime what happen, maybe many of you have also experienced, you are getting very good doc score in computational studies, but when
Yeah,
we are
that's right.
doing experimentally, we are not getting anything, right?
Correct.
So docking score or computational predictions, you have to see sometime lower dock score molecule is showing very good in the experimental part. So you have to look for various parameters when you are selecting a molecule for animal studies or for experimental studies. Because experimentals are costly. In experimental, you can't do Western blot for all the possible
Molecules
target
what
which
we got.
your
Yeah.
molecule can be hit, right? It's costly process. Antibodies are costly. So, and it's also not like that. Depending upon the software is telling you this is the first rank molecule, this is the first rank target, this is the second rank, this is the third rank, based on betweenness or all these parameters. You have to apply your mind as well. Okay, see if one molecule is interconnecting with a lot of other targets, so it could result in various adverse drug reaction as well. There are dual part. So depending upon the ranking given by the software, don't pick the molecules. Of course, sometime whatever the software is telling first rank, by your knowledge or by analysis, you are also agree with that software. Okay, this is the first rank is okay, but you can't go blindly. The software is telling me this is the most prominent target. No, otherwise your experiment will fail. Got it, sir? I
I got it,
hope
sir.
I.
So I'm not. No, no, I'm not speaking about the selection of molecules based on the dark score or something. Assuming
Mhm.
that I'm constructing a network for a molecule for a particular disease. Okay, and I find that it is acting through some pathway or something. So in that
Mhm.
case, so should I go for only animal, or only animal study will support, or should I go for cell line study? That's my question.
Yeah, sir, it depends. Means particularly by doing the in vitro studies, you can screen large molecules,
Yeah.
right? So if you have a large means the number of molecule is more, so your in vitro studies will help you. Otherwise, your animal ethics committee will not give you lot of number of animals to do the
Now, the
these
reason
kind of.
for this question is sometimes back I had a discussion with other experts in the field. So
Mm-hmm.
like I proposed to go for animal studies, and again the issue raised was like, already since you are going in network at the gene level, if you are going at
Mm-hmm.
the animal as such, so
Mm-hmm.
it may not support as such at the genetic level or at the pathway level. So better to go for the cell line studies. So that's why I asked.
Yeah, that again, sir, it's means depend upon how many molecule you have, and particularly you have to see that particular target is expressed in animals, that particular animal species or not,
Correct, correct.
right? If suppose that a target is not expressed in that particular animal, so how you will detect it, right? So depending upon, and of course in vitros you are using human cell lines, human cell culture. So there is a possibility is much more to translate it to the in vitro studies. But of course, means as far as we have done work in animals, we have got the response. But again, selection of target is very important.
Okay,
Earlier,
sir.
our experiment has failed. We have selected the molecule blindly, depending upon the rank, which the software has given the results or p-values or these kind of stuff. But when we did the animal experiments, it's failed. We didn't get any response, even though that target is expressed in the animals, but we didn't get what we are expecting. So then we have means discussed with many experts as well. Then they let us know that the means you can't do it blindly. You have to be see because your lower level the software showing it's third rank, but it's your most important target for these particular conditions, and particularly depending upon their parameters.
Got it, sir. Got it. Thank you.
So you are
Right.
suggesting all next to computational approach, you are suggesting to go for both in vitro as well as in vivo studies, right?
Yes, sir. In vitro, you can do a lot of molecule screening at preliminary levels, and depending upon that, then you can do the animal studies. But nowadays, guidelines, these regulatory guidelines, if you are using 3D cell culture techniques, so if you are showing your effect in the 3D cell culture techniques, so you can skip the animals as well. So there is no need of
Okay.
animal experimentations. And
That
US
similar kind of
FDA has.
results from 3D bioprinting studies we will get, sir.
Yeah, it's a means something where they are means the limitation of cell line is because our system is isolated, and what we are not isolated, our kidney is not isolated. So that is one of the
Okay.
limitation of in vitro cell culture, right? So what the now with the advancement, what the scientists have created, they have created 3D cell cultures where they are creating just like a particularly these human on chips. So if you are using these advanced in vitro cell culture techniques, so there is no need of animal studies.
Okay.
Means I am telling you with respect to regulatory perspective, you can start a clinical trial directly.
Yeah. Fine,
Right.
sir. Fine. Thank you. This is the reason I think you told theoretically explaining all these things are easy, but practically it is very difficult to interpret the actual results to conclude the results, right?
Yes, sir. The problem with students, sir, they are not reading nowadays. So they are thinking, okay, with the help of computer, we can do it easily. But when because for these techniques, you have to read much more detail about
Right.
signaling. Then it will give the confidence.
Definitely, sir. Thank you. Thank you, sir. Thank you very much.
Thank you so much, sir.
Sir,
Sir,
so
one
like,
question is from my side.
mm,
Yes, go
Uh,
ahead.
Yeah, sir, actually, I wanted to know that as far as network pharmacology is concerned, just you told everything that a person just who is approaching network pharmacology, that person must have some sort of knowledge of genomic data, bioinformatics, these things. But sir, as
Yes.
far as a lot of papers just I have seen actually in network pharmacology, they are just targeting mainly the signal detection and pharmacovigilance approach also. So, sir, is
Mm-hmm.
it necessary to have the knowledge of well-versed knowledge of signal detection before going for the network pharmacological approach, or just we can have only the information of bioinformatics, genomic data, and these things, sir?
No, no, no. You should have particularly in which area you are working. So depending,
Okay, sir.
it's not possible to know each and every signaling which is involved in each and every disease. You can't do
Okay.
that. But
Okay, sir.
your area is defined. You plan to work in diabetes, metabolic disorder. So you should know each and everything about metabolic signaling. Then only you can use network pharmacology in a better way when you are doing the analysis of network and other things. Then only you can do it. And to know each and everything about your area, that is required. That is a must. you know
Okay, means here signaling means that detection that we used to do in pharmacovigilance, sir.
no, 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 sir, that signaling is different. That
Okay,
signaling
okay,
is different. I am talking about intracellular signaling. After
okay,
binding
sir.
of the
Okay,
your endogenous molecules, when it bind with a particular receptor and what signaling it going to, like PI3, AKT signaling, mTOR pathways, all these
okay,
pathways you have to know. That signal is with respect to pharmacovigilance. That signal means new things which is coming from the market. If you are planning to use these in a pharmacovigilance, that means that signal is the new ADR which is not earlier reported. That we called a signal.
okay. That is signal detection, sir. Right.
Yeah, that is a signal detection methods, and in that
Okay,
method
okay,
there are
sir.
a statistics we are applying a frequency based method and Bayesian methods, and we are identifying the novel signal of the drugs as per regulatory authority. There are a cutoff values for that,
Okay, sir. Okay, sir. Thank you, sir.
and
Thank you so
this
much.
signaling is about intracellular signaling pathways, right?
Yes, sir. I got like apoptosis pathway, these things like
Yes,
this.
yes, yes, definitely,
Okay, sir.
right, sir, right,
Okay, sir.
right.
Okay, participants, whatever question you have, just you can ask, sir. So just we will be.
Dr. Anoop, Dr.
Yes,
Anoop,
sir.
this is Dr. Sabi from this faculty. I'm working for Ramaiah only. It's
Okay, very good afternoon, sir.
splendid talk, actually. Yeah, very good afternoon. was a splendid talk, actually. Thank you so much for your insightful presentation. And I really appreciate your unique standpoint. At the same time, I have some, view that, suppose the youngsters who are interested to do the network pharmacology, especially and the PhD research, since I'm a budding supervisor for, guiding them. Is there any training program you are providing from your side for network pharmacology?
Sure, sir. Sure. Sure, sir. Means some workshop I can do. Means where students can be with their laptop, and then
Mhm.
it can be done by step by step.
Okay, fine, fine.
There is no issue, sir. And thank you so
One
much,
more
sir, for
question.
joining. Yes.
Yeah, sure. One more question. How can the machine learning be applied to enhance the network pharmacology?
Yeah, for this shortening of your targets, which we are doing manually. So if there are students from artificial intelligence, they can create some tool by which the detection of duplicates or these particularly disease targets and data mining from the literatures. Like Rian is one of the AI based tool. Nowadays we are using. Earlier we are doing it manually, right? So there are a lot of scope where AI or machine learning can play a significant role to ease our process. Because I am not expert in machine learning or AI, we are
Okay.
just a user, right? So I think Ramaiah University have multidisciplinary, courses where many students are. I know some are working on machine learning or these kind of stuff as well. So they can help.
Okay. Okay. Thank you so much, Dr. Anoop. Thank you.
Thank you so much, sir, for joining.
Welcome.
Participants, actually, the link, feedback link is already there in chat box. Kindly fill that form so that just you get your participation certificate later on. And thank you so much, sir, for this insightful presentation. Now, I would like to request our HOD sir and convener of this webinar, Dr. J. Anbu sir, to please propose a vote of thanks to our speaker.
Yeah, thank you, Piyush. Good afternoon to all the participants and today's presenter, Dr. Anoop. And actually, myself, J. Anbu, Professor in the Department of Pharmacology, Faculty of Pharmacy. And it's my honor and a privilege to conclude this session by extending a vote of thanks. First of all, we are very much thankful to our Ramaiah management and Honorable Vice Chancellor Dr. Kuldeep Kumar Raina and Pro Vice Chancellor Dr. Karbanda for their continuous encouragement and support in conducting this kind of scientific programs. On behalf of all the faculty members of the Department of Pharmacology, I thank our esteemed Dean, Professor S. Bharat, for his valuable guidance and support. And we are really fortunate to have an eminent speaker at today's webinar, Dr. Anoop Kumar. In spite of his busy schedule, he agreed to share his scientific knowledge in the area of network pharmacology. We are very much thankful for your support and timely acceptance, sir. And it is a very informative session, sir. And actually, I hope all the participants are very happy with your seminar presentation. And I would like to convey my sincere appreciation to Mr. Piyush Vatsa, for today's webinar's coordinator in identifying you as a speaker for today's webinar. And also, I would like to thank our department faculty members and all the department PG students of our department and club members and the Panpharmacon club members for their support and cooperation throughout the arrangement of this webinar. And my special thanks goes to Mr. Damodar Naik for supporting our department in organizing this kind of webinar and arrangements for today's webinar also. And on behalf of Department of Pharmacology, Faculty of Pharmacy, I profusely thank all the participants who attended today's webinar for their active participation and continuous support. So thank you very much for all the participants. And my special thanks finally once again goes to our Dr. Anoop sir for accepting our request for as a resource person today. Thank you, one and all.
Thank you so much, sir. It's my pleasure to be part of this, and thanks to all. It's nice interaction to all of you. Thank you so much.
Thank you,
Have a
sir.
great day. Thank you.
Thank you, sir.
Sir, actually, as far as our chat box is concerned, actually, we are getting a lot of text regarding the training program for this particular database and all, sir. So definitely we will try to uh, invite you here in future also, so that just we can provide some training program related to this network pharmacological approach, the next generation research mode in future, so that our students can definitely learn and they can incorporate all sort of network pharmacological approach in their paper actually in future.
Sure. It's my pleasure. Thank you.
Thank you so much, sir.
Thank you,
So much.
everyone. Thank you. Have a nice day. Thank you so much.
Thank you, sir. Right.